Um, okay, good afternoon, everybody. This is Nelly Poyak from Dana Farber. Thank you for coming and attending this seminar series. And as just a reminder, the talks will be uploaded on the website. The link is on this first slide, as you see at the FHCC website, and you can look at it, this and all other presentation on that site. And you can have, there's a Q&A on the Zoom link. You can submit questions during the talk. At the end, we will try to answer as many as we can live, but then the speaker will have access to all the questions and she'll be able to answer it later on and get back to you. So it's my pleasure to introduce today's speaker, Kathy Wu, who is our colleague at Dana Farber. She is the Chief of Stem Cell Transplantation and Cellular Therapies, Professor of Medicine at Harvard Medical School, and also member of the Broad Institute. And Kathy will talk about the addressing cancer clonal heterogeneity, developing personalized cancer vaccines. So thank you, Kathy.
Thank you, Nelly. Thank you so much for inviting me to this really awesome series. We're really living in unprecedented times, so it feels good to try to participate in this very new approach to talking about our science. Let me see if I can kind of figure out how to scroll forward. So these are my disclosures. And I'm just going to start off by saying that as a cancer biologist, as a physician, I think as a community, we've realized for some time that tumor evolution poses for us the central obstacle for curative cancer therapy. And we know very well that we've been generating ever more potent targeted therapies, but from the very first moment that we started to embark on cancer sequencing, where we could very well track cancer cell populations on the basis of tumor mutation detection, we could directly observe from human patient samples and even longitudinally over time that there was a high degree of interpatient genetic heterogeneity and moreover intratumoral genetic heterogeneity, and that this was going to lead to a therapeutic challenge. And as you can see in this schematic, what would be a small population initially prior to therapy could lead to the generation of a very different genetic makeup at the time of relapse. And this kind of speaks to the selective pressures that can lead to the emergence of a population that is more fit and lead to resistance. This is not a new concept, but certainly enabled by modern sequencing technology so that we can track it and understand it. And so my group, as many of you know, has been looking at this question from the lens of a particular leukemia, chronic lymphocytic leukemia. And over the years, through large-scale DNA sequencing studies, we have, in fact, been able to track and define the mutational landscape of chronic lymphocytic leukemia, and also look through different therapeutic settings to discern what are the sources of resistance. And I think the takeaway from this co-mute plot, which is at some level superimposable and generalizable across other cancers, is really you can find recurrent mutations, but in fact on a patient-by-patient perspective, each individual tumor has a different profile and panoply of mutations that define it, and really speaks to independent evolutionary events happening within this leukemia. Now, as I mentioned before, the concept of tumor heterogeneity, intratumoral heterogeneity, fueling this concept of tumor evolution is not a new one. Even from the early moments decades ago where scientists like Peter Noel understood cancer as a genetic disease, as one that's really driven by somatic mutation, this conundrum and this challenge to treatment of the therapeutic treatment of cancer was posed. And even at that time, immunotherapy, he posited that immunotherapy would be a leading candidate for the treatment of tumors. And why is that? Well, if you think about our immune system, it's actually hardwired to deal with heterogeneity. It is on a daily, moment-by-moment basis. Even now, as we're facing this pandemic, it's really hardwired to deal with pathogens that are in our environment in all shapes, flavors, and sizes. And also when you start to think about it, you can imagine that if within a cancer there's all these different small subclonal populations, each with a different type of related but different genetic makeup. In fact, one can imagine that an army of T cells actually are uniquely equipped to eliminate that entire tumor population, because it has the potential to recognize a diversity of antigen specificities for each patient per tumor. Now, tumors, we've been thinking about tumor evolution for some time now. Cancer doesn't grow in a vacuum. There is a dynamic interaction with our host immune response, and certainly in the early phases of cancer initiation, one can imagine that cancers can present antigens that can be recognized by the host immune response that could lead to its elimination. But all too often, we're actually dealing with a situation where cancer cells have evolved different mechanisms to evade immunity and inactivate the immune system either directly or indirectly through a variety of different mechanisms. And so it's really been quite amazing. It's not a perfect analogy, but I do think that over the last several years, with the advent of immune checkpoint blockade, we have had our Obi-Wan Kenobi moment where in our fight against cancer, we have a moment where with the availability of these new agents, we have a way to kind of decrease the radar shield of the protective shield of the tumor cells, which is its creation of a negative immunoregulatory microenvironment. And so many of us will remember very well these two papers that came out back to back in the New England Journal that really told us of the dramatic clinical responses that can be achieved with agents like anti-PD-1 antibody for tumors that were never previously thought to be immunogenic. And really, we're in a whole new world right now. It's taken quite a number, several years to get us to this point, but we've recognized with our Nobel laureates of 2018, Jim Allison, Hanjo's work on CTLA-4, and the work on the PD-1, PD-L1 axis with Arlene Sharp, Gordon Freeman, Liping Chen, we recognize that we now have a whole new class of therapy available to us. And many of you know that there is not a single disease center that has not been transformed therapeutically with the availability of these agents, whether or not they're being used as FDA-approved agents or in conjunction with phase 1, phase 2, phase 3 trials. And so, yes, we are at a different time. I think immunotherapy is here to stay as another pillar of cancer care. But even as we recognize that we're in this new age, we have critical challenges ahead of us. It's been estimated that no more than 30% of patients actually do respond to these therapies, and they're not always lasting. And so how can we do better than that 30%? And I would say one way to rephrase that question is to ask, how can we more effectively drive T cells into tumors? And why do I say that? Well, by now there's been a growing number of studies that have linked the presence of immune infiltrates together with outcomes in immune checkpoint blockade. There's been several very lovely studies looking at tumors from the immunohistopathology perspective that have characterized some of these tumors as immune inflamed, linking a high density of CD8 T cell infiltrates in some metastatic tumors that seem to be related to responses to PD-1 inhibition. Also, footprints of that type of response with immune infiltrate in the shape of immune signatures have also been identified in other tumors. On the other hand, we increasingly recognize that there are also tumors that seem to have exclusion of immune cells within the tumor site, or even an immune desert. And listed here are just a variety of different mechanisms, perhaps, by which this type of picture happens, whether it's because of insufficient priming, absence of antigens, dysfunctional antigen presentation, leading to immunologic ignorance, presence of suppressive soluble factors or inhibitory immune cell populations, leading to immune tolerance, or even vascular factors, cytokines, and ECM conditions that can pose barriers to migration of T cells into tumors. And so I guess the question we ask ourselves is then what modalities can we use to drive T cells into tumors? Now, for any sort of complex sort of problem, it's helpful to break it down into different steps. And I think that if we accept that we're doing better in step 2, which is overcoming immunosuppression by virtue of having available to us these immune checkpoint blockade agents, then perhaps what we should be doing is focusing on step 1, which is how can we better, more potently sensitize against the tumor? And there are a variety of different approaches that one could take. So for example, there's pharmacologic agents that may achieve this. There may be cellular therapy. But for the purpose of this talk, I'm going to focus on vaccine strategies because this is certainly one potent approach that can actually lead to improved anti-tumor recognition. And just to kind of get everyone on the same page, when we think about vaccines, typically, and again, especially in the current time, we're thinking about preventive or prophylactic vaccines in the setting of pathogen infectious immunity. So yet what we're facing, certainly in the future, I hope that we can get to this point for cancer. But for the most part right now, we're dealing with treating patients who already have cancer, and hence we are dealing with therapeutic vaccines and the idea of actually stimulating against cancer-specific determinants so that we can increase the breadth and diversity of the T cell response. And so conceptually, what our group has been thinking about for a long time is that if we currently have great reagents in the clinic that allow us to release the brake, we have potent adjuvants that help to accelerate the immune response, can we then not yet devise cancer vaccines that can help steer the immune response in the direction that we want so that we can expand and broaden the T cell repertoire of tumor-specific T cells and thereby generate highly specific anti-tumor immunity while engendering fewer side effects against vital tissues. So that's been kind of our goal. If we're going to generate a cancer vaccine, how can we select which antigens to use to be targeted by the vaccine? I'll remind everyone that when we're dealing with cancer, in the same way that the next generation sequencing has given us a wealth of information about mutations, DNA mutations that have led us to understand more about cancer drivers and pathways, this has also given us a therapeutic opportunity because for sure some of those mutations can be translated, transcribed and translated into peptides that have altered amino acids. And at least some of those, as they get processed, have the ability to bind to class 1 HLA and class 1 or class 2, class 1 HLA and be presented on the surface where those mutated peptides can be recognized by T cells. And these we call neoantigens. Now, neoantigens have been, that concept has been around for a long time, and it's always been thought that tumor neoantigens would be the optimal tumor antigens to go after. Why? Because by virtue of being born of mutations, they have exquisite tumor-specific expression. And akin to pathogens, there's a lack of central tolerance against the targeted antigen because these are somatic events. And so this is really quite different than where we've been in the previous decades, where we've been really thinking about shared overexpressed tumor self-antigens that are variable in their tumor-specific expression and variable in terms of their immunogenicity. What's been super exciting over the last decade with the availability of next generation sequencing is suddenly we have a way to find tumor neoantigens, because until this time it's been very hard to conceive of how one would look on a personal somatic mutation profile basis from individual to individual, how could we find those personal tumor neoantigens? But now, again, with whole exome sequencing available, one can conceive that one could take tumor material, germline DNA, sequence these in parallel, identify those somatic mutations, and confirm by RNA sequencing of the tumor that those actual mutations are actually expressed. And then also, based on HLA typing information, use prediction algorithms that have been available for some time and predict what are binding peptides to personal HLA molecules. And in this way, come up with a list of candidate neoantigens personal for that individual, from which this could be combined with the adjuvant to generate a personalized vaccine. And this concept is certainly very appealing. Before actually getting to the this kind of therapeutic direction, we certainly wanted to review the evidence and assess whether or not this concept actually had some legs. And I think what's certainly been very exciting over the last couple of years, if you've been following this literature, is that in fact, through analysis of sequencing data and immunologic data, by now there is actually growing compelling evidence that neoantigens do appear to be effective tumor rejection antigens. In general, this evidence has come to us in three general buckets. The first is to assess, to say, well, okay, if neoantigens are immunogenic, shouldn't it be that if a tumor has a higher neoantigen load, those patients with those tumors should fare better clinically in terms of outcome than those tumors with lower neoantigen load? And certainly there's been a number of studies looking at TCGA data or specific cancers or following checkpoint blockade therapy where there is some sort of survival benefit or outcome benefit that is discernible. Another bucket of evidence says, well, okay, if neoantigens aren't immunogenic, shouldn't it be that we can go back and look at different settings where there is clinically evident anti-tumor immunity? And shouldn't we be able to see that there is actually evidence of expansion of neoantigen-specific T cells that would signify that these cells actually participated in that anti-tumor clinical response? And certainly my group has looked in the setting following stem cell transplant. The group of Steve Rosenberg has looked following TIL therapy. Various investigators have looked in following checkpoint blockade, and this picture, in fact, seems to be present. Finally, to be a bona fide tumor rejection antigen, shouldn't it be that neoantigen-specific CTLs should actually kill tumor cells, whether in vitro or in vivo? And in fact, there's been very beautiful work both in mouse and man to actually demonstrate this. This is work, again, by the group at the NCI led by Steve Rosenberg, but also mouse models by Bob Schreiber and others. So altogether, we're really at this exciting time and truly a paradigm shift where for the first time, because of the availability of tools such as exome sequencing and also improved algorithms such as neural network-based prediction algorithms on a personal basis, we can identify neoantigens that are tumor-specific and at a personal level, and suggesting that targeting these neoantigens should provide you with a way to gain tumor specificity and lack of toxicity. So can we actually stimulate neoantigen-specific immunity in patients? And this was a direction that my group was very interested in taking over the last several years. And at this point, we have actually completed our first wave of proof of concept studies, which I'll summarize shortly. In the first study that we conducted, and this was together with Patrick Ott at the Dana Farber Melanoma Center, we thought long and hard about what would be the most optimal setting to try to test such a vaccine. And because of the high mutation rate of melanoma, we thought that high-risk melanoma would make perfect sense. And so what we did was we sought to generate a vaccine that would comprise of up to 20 personal neoantigens, why 20? Because we wanted to mount a multi-pronged attack 20 because Craig Slinghoff had already shown that up to 15 peptides pulled together was safe and tolerable in patients with melanoma when given as a vaccine, not neoantigens, but other type of antigen peptides. And because we had kind of modeled based on existing prediction algorithms that selecting 20 would give us anywhere up to 2 to 5 or more neoantigens that we would hope to see were immunogenic. These were formulated as synthetic long peptides, meaning not the 8, 9, 10 mers that we typically think of for targeting class 1, but rather 20 to 30 mer peptides on the basis of work by Case Malif that had shown that this kind of longer peptide format would require professional antigen presenting cells, such as dendritic cells, to take up, process these peptides, and to mobilize and coordinate not only a CD8 response, but also a CD4 response, which we think is important for coordinated anti-tumor immunity, and finally together with a potent adjuvant poly-IC-LC. And our approach was to take patients who had surgically resected tumor from which DNA and RNA sequencing could be performed. With HLA typing and prediction, we could identify those 20 targets to go after, from which these would be synthesized as long peptides, divided out into 4 pools to decrease antigenic competition. And then this would be combined together with poly-IC-LC and given as injections subcutaneously in a non-rotating fashion in a schedule that was prime and boost. So early antigenic exposure up front, and then boost to stimulate immunologic memory akin to what we think is going on with a viral infection. And for this first proof of concept, we were delighted to see that, in fact, when we started to monitor the immune responses following the prime and the boost, we could, in fact, see very potent responses directly ex vivo from our patient. So this is an example where this is a patient who had peripheral blood that was collected at all these different time points. We exposed the blood to these different pools of peptides, and I think what you can see from these different colors is that, in fact, there were new responses coming up that were not there before vaccination, and you also see the different colors. So our goal of having a multi-pronged, multivalent response, in fact, already seemed to be quite evident even within these first experiments. And across the 6 different patients that we treated on this study, as we started to deconvolute out what was underneath each of these pools, in fact, we could confirm that there was actually responses that were specific for the mutated peptide, not against the wild type when testing against a range of peptide concentrations. Because, and I think what I want to emphasize that certainly as a first step to look at what's in the peripheral blood is good. It certainly is a measure of the immunogenicity of the vaccine, but ultimately our goal is actually to target the tumor. So the other key experiment that we were focused on performing was actually to go back to the original tumor from which we had generated cell lines, and then to test actually the neoantigen specific T cells against the autologous tumor. And what's shown here is certainly for some patients, we could definitely demonstrate that there were both CD4 as well as CD8 T cells that we could demonstrate against autologous tumor. And in this instance, this interaction could be blocked with class 1 blocking antibody. You see that there's actually minimal response in the setting of testing against irrelevant melanoma, and really speaking to the personal nature of the response. These 2 are ELISpots, so these are both assays that allow us to detect response of T cells on the basis of interferon gamma secretion, which is indicated by these dots. Now, how did these patients do? This was a small study, 6 patients, and I'll mention that 4 out of the patients had stage 3 disease. 2 of the patients had resectable stage 4 disease. And perhaps, and so at the time that we reported this, all patients had received vaccine shown by these blue bars. And at 25 months, patients were doing well. Not unexpectedly, these 2 patients, patient 2 and 6, shortly after their vaccine recurred and that's shown by the orange triangles, and their treating physicians then started them on anti-PD-1 antibody. But what was pretty exciting and interesting is that after a short, about 12 weeks of therapy, both of them achieved complete clinical responses that have since endured. And this is shown on the images here for patient 2 and then patient 6. You can see that there were antecedent lesions before starting the anti-PD-1 therapy that then regress after initiation. And this was, again, small series, 2 patients, but I'm told by my melanoma colleagues, clinic colleagues, that in this setting that responses to anti-PD-1 therapy are in the range of 5 to 15%. So certainly we were excited to see this, and really, if you think back to the cartoon that I showed earlier, this idea of a 1-2 punch, having vaccine together with PD-1, anti-PD-1 antibody, was certainly exciting to us, and something that we're excited about pursuing further. Now, no good idea ever happens in isolation, and certainly at the same time that we were conducting our neoantigen vaccine trial, similar efforts were underway by our colleagues led by Uber Sahin to test such a vaccine again in high-risk melanoma. And so the design of their study was different in the sense that not only did they vaccinate with against personal neoantigens, also discovered through a computational approach, but while the manufacture was going on, they also vaccinated with against a shared tumor antigen. A key difference is that whereas we used peptides, they used RNAs as a way to deliver their vaccine targets. And I think what both technologies share is the way to kind of nimbly slot in and slot out the composition of your vaccine. And so, as I mentioned, so we used peptides, they used RNA. Our epitope blends were a little bit different. Whereas we had 13 to 20 in our vaccine, they targeted 10. But I think what's pretty interesting is in terms of the type of T-cell responses that we both observe, they're relatively on par with each other. And I think this really points to the idea that there are multiple different delivery approaches that can get you to the same place, and also reinforcing the idea that a genomics-guided approach can in fact identify immunogenic targets consistently across different centers. Now, I don't think it's by chance that both our groups targeted melanoma as kind of the first stop, and it does make you wonder, is this approach only applicable to tumors with exceedingly high mutation burdens? And so the question that we asked is, well, what about a lower mutation rate tumor? And our group had the good fortune to work closely with David Reardon in the Neuro-Oncology Center, and you can see from this kind of somatic mutation chart of tumor burden across different cancers that glioblastoma is much lower in mutation burden than melanoma. But as we started to think about these patients with glioblastoma, there were some appealing aspects to testing the vaccine in this setting. So for one, these are patients who typically get surgically resected. That's what leads to their diagnosis. And following a period of recovery, those patients then go on and receive several weeks of radiotherapy. And so already built in here is about 8 to 9 weeks of standard therapy for which one could imagine to taking that surgically resected specimen and going through the same process of vaccine generation so that this can be given in the back end in an effort to stimulate a immune response in these patients as they recover from radiotherapy. And so our question was, one, can we hit lower mutation rate tumors? And second, how could this be incorporated into together with standard of care therapy? And many of you know glioblastoma multiforme is an awful cancer. It's really very aggressive, rapidly fatal. It's been thought to be a cold tumor. So not a tumor where there's not a lot of T cells that are able to get up there. There's been questions about blood-brain barrier. There's already been observations of suboptimal responses to anti-PD-1 therapy alone. And so certainly a very, very challenging setting to go after. But nonetheless, we were able to enroll 8 patients onto the study, which are reported here. Just to lead you through this timeline, as I mentioned before, these were all patients. The timeline starts with surgery. As shown in the yellow bar, they all go through radiotherapy, and then the arrows on the top indicate when in their course they received vaccines. So again, priming and then followed by boost. One of the disease-specific things that we realized about this specific setting is that actually many of these patients are receiving corticosteroids, and this is receiving dexamethasone. Corticosteroids is almost second nature to patients getting treatment for GBM. It's such a constrained space, and really to kind of with concerns of cerebral edema, these patients will often get very, very high levels of dexamethasone. So this shown here is the color bar. You can see the darker color, the higher dose of dexamethasone. And if you look across the patients, you'll see that there was a lot of dexamethasone on board. But I do want you to take note that patient 7 and 8 were the 2 for which there was not actually dexamethasone on board when they received vaccine priming. So just remember that, because this will come back later on. The other thing about this setting that actually attracted us to working in this is that it is also reasonably common that when there's a suspicion of progression, so here shown by the X, that the neurosurgeons typically go back in, and there's the opportunity to collect a second specimen, which we could study as well. And so you'll note here that there were 5 patients for which there was a second surgical resection, for which we had tissue that was collected after vaccination that we could examine. So our first stop was to look at the peripheral blood responses. At the same time that we were seeing fantastic, strong, potent responses in our patients with melanoma, for many of the patients that we monitored, it was really very little by way of response, and that's there's a bit of a response with patient 6, but this is really due to high background. So really there was really minimal absence of response that we saw in our peripheral blood monitoring, and that's when we realized that these patients, for the most part, were receiving dexamethasone. By contrast, patients 7 and 8 were both those 2 patients who did not receive dexamethasone. And what you can see here nicely is that, in fact, they generated immune responses on par in strength with what we saw with our melanoma patients. I won't lie, this was a much harder setting to design our vaccines. There were much fewer mutations, and we had to work a little bit harder to compose the composition of our vaccines, but we were able to successfully get there, and you can see here that these were the responses that we were able to pull out in patients 7 and 8. The other interesting thing that we could see was that actually when we started to look at those 5 patients who had initial surgical specimens and the relapse specimens, that as we started to look, we asked, well, do we actually see evidence of T-cell infiltration? And it turns out that those 2 patients who hadn't received dexamethasone, patients 7 and 8, compared to the 3 that did, we actually did see significant increases in CD4 and CD8s that were discernible on sections. And that's shown here, where the CD4s are in yellow, the CD8s are in green. And having seen this type of infiltration, we began to ask ourselves, well, maybe the vaccine is actually leading to a warming of this cold tumor. Would it actually be, what are these T cells actually seeing? Are they actually reactive against the neopeptide? And how could we figure that out? And so, like in any sort of human experiment, I think we're always confronted with challenges. We have to ask what kind of material we have. And so for patient 8, we certainly did have cryopreserved peripheral blood mononuclear cells, and we also had fresh frozen FFP material. And what we realized is actually, when you think about T cells and the T cell receptor, tremendous amount of diversity in the TCR, which is defined by both the CDR alpha and beta chains. And hence, the TCR actually ends up becoming a barcode of sorts of T cells. And so then so with that idea in mind, we devised an approach where we thought to ourselves, well, okay, in patient 8, we already know that this patient is reactive against pool B, and we've deconvoluted, so we know that patient 8 is reactive to these 2 antigens, SHANK2 and SVEP1. So if we take blood and we actually stimulate against these antigens that are neoantigens that for which this patient is reactive, and then we perform TCR sequencing, could we then actually identify what are those clonotype information of those neoantigen-specific T cells? And then if we know that information, then maybe we can go back directly into the brain tissue, and then through bulk TCR sequencing, look to see if any of those same molecular sequences are present and enriched. That's what we did. We took blood following vaccination. We stimulated against these 2 mutated peptides. We were able, on a single cell basis, to characterize the TCR sequences, and this is basically the map of what we generated after this procedure. So for MutShank, you can see that each dot represents 1 T cell, and if they're next to each other, they're actually the same clonotype. So for both MutShank2 and MutSVEP1, we can see that there are expanded clonotypes where we know that these direct TCRs are related to, are recognizing these mutated peptides. And so the actual sequences are shown here. And then with that information, then we were able to go directly into the brain FFP mRNA. And in fact, you can see that for one of the mutated peptides, but not the other, we could actually track T cells that seemed to be specific against MutShank2 directly in the brain, both the alpha and beta chain. And this is already super interesting because this is telling us that actually these T cells are actually able to get to the tumor site, but there's also differences between antigens in terms of their ability to do so. Now, what about patient 7? Well, patient 7, we actually had the good fortune to work closely with Mario Suva and Kai Wuferfenig, and together we were able to actually in real time obtain tissue and gather, select out T cells. And so directly from the relapse sample, we could actually perform single cell transcriptome profiling. And because we had those single cells from brain tissue for which we could again perform TCR sequencing, we decided to then also perform the same approach from the peripheral blood side, which is, again, knowing that this was a patient who had reactivity against pool C, which was deconvoluted to this RGAP35. There were responses both for CD4s and CD8 cells. And then we wanted to ask whether or not there were clonotypes that were in common between the two. So in other words, T cells, TCRs that directly recognize these antigens that overlapped with TCRs that were directly in situ from the brain. And if so, could we then reconstruct those TCRs? Because we had just reported out an approach where we could rapidly clone out T cells based on their CDR3 alpha beta chain information, essentially freezing a TCR, getting it into a readout cell line. And in that way, then we could interrogate directly the specificity of those T cells. And so again, this is what we did. We took the patient 7 tissue, the CD3 cells were selected. We were able to generate not only a transcriptome libraries, but we were also able to perform targeted TCR sequencing, and from there perform TCR reconstruction. So here's what we saw from the brain tissue. In the orange are the CD8s, or in the blues are the CD4s. So already we see some very fascinating sort of clonal architecture amongst the T cells that are infiltrating in the brain at the time of progression for this patient. But then we also see that if we take the peripheral blood samples that have been stimulated, we can find that there are clonotypes there. And what's fascinating is that we in fact did find that there were some clonotypes that were in common between these two compartments. And upon reconstruction and expression and testing of those 6 TCRs, 2 out of the 6, in fact, we could confirm were reactive against RGAP35, which was in the vaccine. So here we have direct evidence that what we are stimulating peripherally on the limbs as a vaccine is generating T cells that have neoantigen specificity that are actually able to track to the site of the tumor. This was super exciting. What was also cool is that because we had single cell transcriptome information about these single cells, we could also track to see what was the state of those cells underneath those specific TCRs. So here you can see that the HO2 TCR, which is shown in the red, the F10 TCR, which is shown in the green, you can see that both of these have transcripts consistent with markers of cytotoxicity, like cytostatin, granzyme A, granzyme K. One of them expresses more IL-2, has more transcripts for IL-2. The other one, more transcripts for TNF alpha. And then whereas one does not express any of these co-inhibitory receptors, the F10 is expressing transcripts for PD-1, TIGIT, and TIM3. So already in the story of 2 TCRs that are targeting the same neoantigen, we already see heterogeneity in terms of their kind of transcriptional state, and also giving us clues about what kind of co-inhibitory receptors are operative in this type of microenvironment, and certainly food for thought as we think about how to build combinatorial strategies for GBM in the future. And so just to summarize this part of the talk, I just want to say that we've gone from showing the feasibility and the immunogenicity of neoantigen vaccines that are being built on a personal basis. In the melanoma site, showing that it's highly immunogenic and in conjunction with checkpoint blockade, even showing that there's exciting clinical responses that we can see. In the GBM story, we've observed the adverse impact of steroids with T-cell priming, leading us to think about the design of future clinical trials. I hope that I've convinced you that neoantigen-specific T-cells can variably track to the site of tumor, and that they have variable transcriptional profiles. And we, again, really look forward to combination with checkpoint therapy for this disease in the future. And just, again, as we think about what we need to do in order to get to cytolysis of the tumor, at the minimum, we need to demonstrate that we have activation and proliferation of tumor-specific T cells, which we have achieved. We need migration to the site of tumor. We're on the road there. I think we have much more that needs to be done, but at least I think that we're moving in the right direction. These type of first wave studies have led us to generate a second wave of studies that are beginning to open up at Dana Farber. We already have an exciting study that is ongoing together with Tony Chuari in the renal cell, in the GU group for clear cell renal cell carcinoma that has also been co-led by David Braun. And we have a number of other studies that are planning to be open across these different disease centers in 2020. So we really look forward to the insights that we can gain in all of these very different disease settings, each one that I think will help us learn more about how we can better deploy cancer vaccines. But a critical effort in our group has been to think about how we can build better vaccines, and not the least is the thought of how can we do better at prediction, because part and parcel of our approach is actually starting with the genomic data so that we can make good choices about what to put in our vaccine. Now, I want to remind you that the current prediction algorithms that are out there and have been out there for some 15 years and ever getting better, have in the past really focused on this terminal aspect of what happens with antigen processing presentation, which is just binding in the very end. And actually, it's been really quite a black box in terms of what's happening within the tumor cell in terms of processing. And so this is a question that we wanted to get at, because certainly if we could understand this aspect better, maybe we would have a better clue about what actually gets to the surface and is presented and visible to the immune system. And so for quite some time now, together with our colleagues in the Broad Proteomics platform, we have been thinking about how we can actually use mass spec as a way to directly learn what is on the surface of tumor cells that are bound by HLA. And one approach that has been used to study the immunopeptidome is a general multi-allele approach. And a typical approach would be to either take a cell line or a tumor specimen where you have, at least for class 1, you have 6 different alleles. You would perform a IP with a class 1 antibody. Typically, this requires a lot of cells, and then you would elute off those peptides, and this would go through a mass spec instrument. And the first step in a lot of those steps is actually, in terms of the analysis of the spectra, is first inference of the binding allele assignment based on pre-existing predictors. What we wanted to do was actually to engineer, control the situation a little bit more and actually engineer HLA balled cell lines so that they would only express single HLA alleles. Also, a series of optimizations had been undertaken, led by Darren Keskin in our larger group, and really thinking about how we can actually get to lower amounts so that it's actually feasible to actually process a lot of samples in parallel. And in that situation, all the peptides that come off, you actually already know unambiguously what the allele assignment is. And hence, from the data, you can de novo learn the rules of antigen processing and presentation in relationship to specific alleles. And this is the approach that we already reported out a number of years ago. And at that time, we looked at 16 different lines. We were, in fact, able to demonstrate that this type of mass spec-based immunopeptidome approach could lead to better predictive algorithms and could be conducted in high throughput. And so most recently, again, spearheaded by Darren Keskin, we have actually reported out data collected for 95 different HLA alleles. This is more than 180,000 peptides that we have identified and across different alleles selected, so that in aggregate we have 95% coverage, global coverage over the world population, and this more than doubles the number of positive observations in existing databases. So this is a huge wealth of data covering a lot of the different alleles. And with this type of data, then you can start to look at super interesting questions. So for example, we can begin to look at the differences between HLA-A, B, and C. And so this is just an example of an analysis that Cici Sarkazova, a recent doctorate student in near Hakohen's lab, generated. And really, we could look at distinct motifs that were identified and clustered based on allele-specific submotifs. And I think what you can appreciate is that per motif, there are contributions across. You could see that this is covered across many different alleles. And this definitely raises the question that given that there's overlaps and submotifs across alleles, this would lend itself for the basis of optimizing epitope selection such that a minimal set of epitopes that can cover an optimal set of multiple alleles and thus individuals can be generated. again, when we're looking across the different alleles, we begin to see differences in length preferences. So for example, for HLA-A, there seems to be a preference. We're typically thinking about 9 mers for class 1, but there does seem to be a length preference for the longer peptides. And for HLA-C, for the more for the shorter peptides, whereas for HLA-B, it's really variable. And where this became interesting is that as we started to look on a per allele basis, that there are some peptides that whether it's a 9 mer or a 8 mer or a 10 mer, the actual binding motif changes. So examples are shown here. So for example, this is, if this is the 9 mer for A33, you can see that the motif for the 8 mer is really quite different. The same goes for this example of a B14, where we see, again, subdominant motifs that are anchors that are really quite different, depending on the length of the peptide, and so on and so forth. And so these were some of the insights and differences that we saw that we could then build in as CC started to take into account all of these different variables and started to build new neural networks and integrating all of this mass spec data. So that's noted here, MSC for cleavability, and then EC for expression and cleavability. CC was able to generate both allele and length-specific models, as well as use the data to train pan-allele, pan-length models. Now, one of the things that comes out of doing this type of work is that there's a lot of modeling that goes on, but then you ask, well, is it actually making a difference? And so what we did was actually generate a data set of peptides eluted directly from tumor specimens, and this became our observed data set. And then we asked if we actually then tested against all, not only our mass spec-based models, but existing models out there, how does our prediction model perform? So in other words, how does our prediction fare compared to what we have already observed in the observed data set? And it turns out that our new tool, which we call HLA Athena, actually definitely performs better compared to the existing NetMHC pan models. And in particular, we see that there's 9 to 38% increases in peptide identifications. And this is really quite meaningful for those lower mutation burden tumors where, as we saw already with GBM compared to melanoma, where it is harder to identify vaccine targets. Perhaps if we have a better predictivity, we don't have to go so far down the list for us to really identify truly immunogenic peptides. So we're very excited about this, and this algorithm is now being incorporated in our new clinical trials going forward. So I'll stop there. I just wanted to share with you our approach of using a monoallelic strategy to profile HLA alleles that can provide an unbiased view of peptide binding preferences. Our prediction models outperform state-of-the-art predictors in distinguishing tumor-presented epitopes. I want to point out that we've been able to expand our prediction beyond HLA-A and B and now also incorporate the HLA-C alleles as well. And just to conclude, I think that I just want to mention that there's so many other aspects that we need to address in order to make this type of therapy more common. And so certainly, the questions that we have are, how can we decrease the time from tumor acquisition to vaccine administration? How can we understand better the number of peptides, the administration schedule? So these are old kind of vaccinology questions. How can, currently our studies have been in the adjuvant setting. What about, the more challenging settings of metastatic tumor, and so on and so forth. And so these are all questions that we're thinking hard about, and also trying to figure out how vaccines are placed within the panoply of other therapies that are out there. So for example, in the future, if we're able to better immune profile our tumors and understand whether they're inflamed or excluded or immune desert, is there kind of a sequencing and rational combinations that we can think about where vaccines can play a role. And so I'll just end there. I want to really thank all the people in my group who have really contributed to this work. For the 25th anniversary of the journal Immunity, they had asked cancer immunology labs to contribute photos of their group. This is kind of a impromptu photo we took right after lab meeting, and you can see that there's a good portion of folks in the lab who are working on biology of various cancers, but also a large group that is working about on tumors, on T cells, anti-tumor T cells and vaccines. And I also just want to mention that this work would not be possible without being in this amazing environment that we are in. And so certainly in Dana Farber, we've had the benefit of kind of the full support of conducting investigator-initiated clinical trials and the ability to perform characterization of patient samples gathered on those trials, and working very, very closely with our colleagues at the Broad Institute, and also increasingly with our colleagues over at MIT. And so I want to thank you for your attention, and I would be more than happy to take questions. Thank you.
Thanks, Katie, for this amazing talk and work, actually. So we have a long list of questions, and many of them related to your TCR, how good TCR sequencing predicting the neoantigens, and I think some of those you already answered towards the second part of the talk. But basically, many people ask, how can you predict the neoantigen? How can you predict which one is presented the best? How do you pick a combination of these peptides, which, I know all of these are very challenging questions, but I don't know, and some of it you already answered. I don't know if you want to give a brief answer to multiple of these.
I will try, and I just, so this is like, new adventures in Zoom land for me, so I'm trying to look at the chat and figure out how this goes. But in general, in terms of the selection of the neoantigens, I think, we've kind of come up with a schema that incorporates prediction based on the algorithms. We also, at the current time, because we're working, we also look at expression. We also look at feasibility for generating peptides based on their biochemical characteristics. We also consider trying to get coverage across the different HLA alleles in the patient. I think one member of the audience asked about class 2, and this is an interesting question. We really focused our vaccine on trying to go after class 1 epitopes, but in fact we generated a broad, actually a very strong, robust CD4 response, and this was, but we actually confirmed that those CD4 cells had specifically recognized the mutated epitope and not the wild type. So this was very interesting. We don't understand exactly how that is happening, and somehow we're getting CD4s for the ride without even having to predict for class 2. But I do want to mention that there are in this past year a number of very interesting papers, both in animal models and also in the prediction world, of new tools for class 2 prediction. And so we're in the process of testing out those tools and trying to think about if we should incorporate some of that aspect.
So another challenging question from Bill Kalin is what do you use when you're trying to compare the blood and, for example, the GBM T cells, reactive T cells? What do you use as a normalizer? Because, from a technical point of view, because it's possible that you have circulating T cells and a fraction of them get into tissue. So do you use another tissue type as a normalizer to prove that this is tumor specific, or how do you deal with this?
Yeah, no, this is a great question. It's actually, Bill, the answer is in the paper, in the supplemental. but basically, we actually did a calculation of what was the likelihood. So the answer is that the statistically, the frequency to which we detected those antigen-specific T cells in the brain tissue was much higher than the detection in the blood. And we had matched ELISpots for the specific epitope, so we could actually count cells, if you will, that were in the blood that had that specificity and do the normalization that way.
Okay. So maybe we have time for one last question, another challenging one. It's like, how does the vaccination change the heterogeneity of the tumor in terms of the mutational landscape and the neoantigens, and why the tumor doesn't just lose certain neoantigens?
Yeah, and great question. And I just want to emphasize that our data is really sparse, right? So we in the end of the day, we only treated so many patients, and we are also limited by the time points, right, for which we can sample. So the short answer to this very interesting, complex question is that I don't know. we haven't had enough experience to look at that. I mean, we're beginning to start to monitor some of those melanoma patients further out, and being able to look at, for example, local relapses that happen, and being able to look at those genetic changes. But again, these are really, really sparse data, so I'm not sure how much broad conclusions that we can make from them.
Okay, well, thank you so much. I think I know there are many other questions. Kathy will have a chance to look at those after the presentation. So thank you again. It was an excellent talk, very stimulating. And we'd just like to remind people that you can look at the presentation on the website, the link that we posted, the FHCC. And thank you again, everybody, for participating in the seminar. Bye.
Thank you. Bye. Take care. Be well.